Welcome everyone to this evening's BMS Talk, the first talk of 2024. I'll introduce our chair, who's actually someone who probably doesn't need much introduction, but anyway. Lynn Boddy is Professor of Fungal Ecology at Cardiff University. She's a prolific author, having written or edited 9 books and published over 300 scientific papers. Lynn was awarded an MBE in 2019 for services to mycology and science outreach. So thank you, Lynn.
Thank you, Sally. And welcome everybody in the audience. We're looking for lots of audience participation today, please. So let me remind you that we have the chat facility, and please put your questions in the chat. And I think that Mark will be asking you specific things as we go along for you to comment upon in the chat. So we hope to have a very good discussion at the end. But before we get that far, let me introduce to you Mark. And I think I probably first met Mark in the late 1980s, early 1990s, when he was an undergraduate at Exeter. And I was hugely impressed then by his knowledge, understanding and enthusiasm for fungi. And indeed, I'm still impressed today on those same counts. Mark went on to do a PhD with Alan Rayner at Bath, and then spent a brief time in industry, did a postdoc fellowship at Cambridge, and then had several fellowships at Aberdeen before moving to Exeter, where he is now. And Mark researches on programmed cell death in Candida albicans. He's a molecular mycologist. This always impresses me because I'm afraid my molecular skills are really rather negligible. And so Mark is a very strong researcher, both in the lab and in the field, but actually he also spends a huge amount of time and effort on teaching, and I think he's still Director of Education, probably been doing that for 8 or 9 years now. And obviously, Mark teaches undergraduates and postgrads at Exeter, but he also does a huge amount of outreach, including, or ranging from, I suppose, school children to the wider general public, from farmers to medics. So Mark has a huge amount of experience, and today he's going to tell us about insights into fungal education and outreach. And you can gather from that introduction that he's very, very well qualified to do that. So very warm welcome, Mark. If you'd like to share screen now, and over to you.
Okay, well, thank you very much, Lynn, for that wonderful introduction. And I feel as though I'm in very auspicious company in relation to this particular topic, because you've been at the forefront of fungal education and outreach for a very long time. So thank you very much, Lynn. Today's talk is about insights into fungal education and outreach. And I guess I'm taking a kind of reflective review of some of the contributions that I've made very recently in my role as chair of the Fungal Education and Outreach Committee of the British Mycological Society. And that's opened all sorts of possibilities in terms of trying to talk to new audiences and talk about fungal biology. You're going to see quite a lot of slides. You're going to hopefully learn some exciting and new things. I don't know exactly who's in the audience today, and I'm sure there are many people who are very, very experienced in fungal biology and some in education. And so what I want to do is encourage a bit of a dialogue. So I'll be asking some questions throughout the talk, and if you can post the answers into the Zoom chats, then after the session, Lynn and I will go through those comments and use that as a starting point for a conversation. I'm also leaving this slide on the screen for quite a long time at the beginning to emphasize that UK Fungus Day is one of the core activities of the Education Committee of the BMS, and this year's UK Fungus Day will be on Saturday the 5th of October 2024, so please pop that into your diaries now. So here's me, I suppose, reflecting on what it takes to become a mycologist. And from that starting point, I can talk a little bit about my educational inspiration and some of the activities that I'm currently involved with. So I started off with this little animation or diagram showing how a fungus develops. And of course, early on in my career, I was basically like a spore, very small, somewhat insignificant perhaps, and naive to the world. And a little bit like a developing fungus, over time, and I've probably been an active professional mycologist for over 30 years now, I've developed knowledge and understanding of the fungi, a passion for the fungi, and had lots of opportunities to try and pass that information on to others. So like a fungal spore, you have to increase in size, adapt to the environment, change your morphology, maybe switch into a networked mycelial form. That mycelium, of course, can explore the environment, then capture new resources, consolidate that, and then commits to differentiation and the production of a complex multicellular structure, such as a fruit body. The culmination of that, of course, is then the production of new spores and the dissemination of that out into the environment to restart this cycle. So what I want to do is kind of use that as a metaphor for my own development as a mycologist. This is a picture of me taken when I was about 3 years old. And it's very clear to everybody in my family that I actually had a passion for everything to do with natural history from a very, very early age. And for some reason, whilst I was about 5, 6, 7 years old, I became fascinated by fungi, I became fascinated by fishes, and fascinated by insects. And we will revisit those a little bit as we go through today's talk. Here's me in my fishing stage, very proud fisherman, having caught some mackerel on a holiday down here in Devon, which is now where I live. So it's great that I now reside in the place that I used to holiday as a child. And here's me as an undergraduate when Lynn actually first met me. I think this was during the final year of my time as an undergraduate at Bath University. And at that time, I was completely caught by the importance of fungi, the stories that were being told to me by my mentors, and the fascinating role that fungi play in the natural environment. I've cropped out my best friend who's actually in this picture. I'm explaining something really, really important to him, but he looks bored stiff. But this was me as an undergraduate some 30 years ago. And just to recapture the moment, here's me a few months ago to recreate that first image now in my garden, looking at some plant pathogens on plants in my garden. Of course, the inspiration to be a mycologist doesn't just come from their amazing biology. It also comes from their beauty and their diversity of form. We estimate there are somewhere between 2 million to 12 million different fungal species on the planet. That's an enormous number, and they are ecologically very diverse and evolutionarily very diverse. And we have within the kingdom of the fungi, the true fungi, we have a range of different phyla or divisions. And so there are many, many different characteristics shown by the fungi. I just want to explore a few of these here. One of my inspirations has been the awesome power of yeast molecular genetics. Yeast, Saccharomyces cerevisiae, baker's yeast or brewer's yeast, depending on your preference, has been was the first higher organism to have its full genome sequenced. And I've spent many years now trying to understand the role that those genes play in things like stress responses and death responses of fungi, particularly in human fungal pathogens, but using yeast as the model upon which to base those studies. Of course, In my work over many years, I've encountered all sorts of different fungi. So this is the Pylobolus that grows on cow dung. Very nice, simple experiment that I get my undergraduate students to do is to take some cow dung or rabbit pellets, pop them into a yogurt pot, bit of damp paper in there and leave it overnight. And after a few days, you start to see these beautiful sporophores being produced with the sporangia at the top. And of course, these cells are huge. They can be several centimeters long. And when they discharge the spores, they accelerate this little package of spores at around about 20,000 G, which is the fastest acceleration ever recorded for any biological entity. So you can see not just from the biology, but also the beauty, why I've been inspired to be a mycologist, and perhaps you have some of your own favorite fungi as well. I wanted to acknowledge the importance of people in gaining insight into fungal biology and biology in general. And a little bit later on, I want you to think about who has perhaps influenced you to take up mycology or to invest time in studying them. I realized that when I put together this series of pictures of people that have been important to me, many of them emanate from a very formative period in my life when I was about 10 years old. So here we have Gerald Durrell, and obviously his books that I read as a child inspired me. He wrote them as if he was a child, reflecting on his childhood and telling stories about the amazing things that he did in Corfu. His David Attenborough in that famous scene with the gorillas. This is taken from the Life on Earth series, which was released in 1979. We have here David Bellamy, who was a British botanist and television presenter. And I have a copy of a book here called Botanic Man. I don't know which way up that goes. Botanic Man, there we go. From that time, and it still sits on my shelf in my office. I did have a look at it earlier, and there's one short paragraph about fungi in there, which is a little bit disappointing, but you know, some things don't change. Next to that, we've got Heinz Wolff. He was the television presenter lead and actually an education and outreach ambassador, which was quite an unusual thing back in the 1970s and early 1980s. And he presented a television program called The Great Egg Race, and it inspired in me a desire to be very creative and to solve problems. On the bottom row, this is the other side of my interests in biology, which was marine biology. And this is Hans and Lottie Haas, who were explorers in the 1950s and 60s. And I read lots of, as many of their books as I could when I was a child. Moving on, though, there's a row here of people who are mycologists, and they've all played really important roles in my personal development as a mycologist. So as an inspiration, John Webster, who was the head of biosciences at Exeter, I think he might have been the head of biosciences when Lynn was a student at Exeter back in the late 1970s. Here we have Henrique Descaldes, an aquatic hyphomycete expert, and we'll return to him a little bit later on. Then we have Alan Rayner. So I met him as an undergraduate at the University of Bath. He was my personal tutor. I think that was something that didn't quite happen by accident. I think they must have read my university application, and he took me under his wing during my time in Bath. And here is Alistair Brown, who was my mentor whilst I was an independent research fellow at the University of Aberdeen. So all these people have contributed in some way to my passion for fungal biology in its wider context. So this is your opportunity to use the Zoom chat function and just put in names of people, roles that they might have played in terms of what brought you to mycology and your particular interests. Okay, so you can do that. I'll carry on talking. And I want to come back to Henri Descaldes. Henri Descaldes was a postdoctoral researcher in John Webster's lab in Exeter in the early 1980s. And I have a very strange tale to tell, but essentially back in 1980, my parents realized my passion for fungal biology, and they rang up the University of Exeter botany department. They got their number from an old telephone book. They rang the number rang for a little while, and somebody answered the phone. The person that answered the phone was Henri Descaldes, who I have very fondly known as Henry ever since. And Henry essentially had answered a public call box on the University of Exeter campus. And as one of only about half a dozen mycologists in the whole university at the time, that was quite a chance moment. But he understood that I was interested in fungi, and he invited me to the University of Exeter and started training me in laboratory work from the ages of about 12 years old. He took me on collecting expeditions locally in Devon, but also in here in the Pyrenees. And we went and collected aquatic hyphomycetes from foam and from water samples, and also were collecting black flies that had died on rocks just above the water level in streams, and that was part of his ongoing research at the time into these entomopathogens and their potential at that time as biocontrol agents. So very early on, I had an experience of working in the laboratory and also undertaking field work. But I think critically, it was also a training in microscopy and the beauty of things that you can see, that hidden world under the microscope. So here's a summary of some of the mycological training experiences I had leading to where I am now. I did my degree at the University of Bath. I worked for the Forestry Commission on a 6-month placement looking at mostly insect pests, but also working on projects with Clive Brazier, looking at forest pathogens. I did another 6-month placement working for the Ministry of Agriculture, Fisheries and Food, learning biochemical techniques and looking at mycotoxins in food. And then I undertook my third placement with 6 months at the Institute of Terrestrial Ecology at Merlewood in Cumbria, working for Juliet Frankland, working on fungal decomposers of leaf litter. So these very different experiences really inspired me to carry on in a mycological vein. So I applied for a number of PhDs and ultimately chose to work at the University of Bath, which is where I've been for 4 years, working with Alan Rayner on genomic disparity in basidiomycete fungi. After graduating, I needed a job, so I worked for around about 18 months at Pfizer in Sandwich in Kent, and again my primary role was investigating fungi, looking particularly at fungal secondary metabolites and increasing the diversity of secondary metabolites that could go through their screening programs. And then I was a postdoc in Cambridge working on Neurospora crassa, the first time I'd actually worked on a model fungus and the advantages that gives. Working on circadian rhythms, we isolated mutants that had defects in their clocks, so they had slow running and fast running clocks. We were able to use the characterization of those mutants to understand a little bit about the biochemical basis of clock and timekeeping in cells. And then I moved to Aberdeen. So I was a postdoc initially for 1 year working on yeast molecular genetics with Alistair Brown. And then I started applying for research fellowships and obtained one from the Lloyd's Centenary Foundation, which set me up on the pathway to understanding fungal programmed cell death and stress responses. That ultimately then led to securing a lectureship in Exeter. I never specifically chose to go to Exeter. It just happened by chance in many ways. And I've now been in Exeter for 17 years. During my time as a researcher, I was also involved a little bit with education, but my role primarily for the last 10 years has actually been in education activities, both at the departmental level, running the Biosciences Education Programme, and more recently as the Director of Training for the MRC Centre for Medical Mycology. So at this point, I want to change tack and introduce everybody to the current stars of the BMS education portfolio. So I think many of you know Lynn and her background as an educator, but there are other people on the FEO committee with different expertises and different backgrounds. We've tried to bring together people with diverse interests. So it's not just academics, it's people with experience and expertise in the use of social media. So Evelina and Ethan in particular, people with a passion for fungal education, from the foraying angle, so Cameron and Christian. Christian is also a secondary school teacher and Open University lecturer, so he's got a great deal of experience of talking about fungal biology to all sorts of different groups and interest groups. And Gary, who is also an amateur mycologist, based locally down here in Devon, and he brings a lot of insight into the local groups and how they want education to be put forward. So how am I going to talk about this wider subject of science communication and outreach? So I've divided this next section up into the who, what, why, and how. These are all things that we need to consider when we're putting together a package for educational purposes. So we need to think about the audience, the diversity and the backgrounds of the people in that, the age ranges, their prior knowledge and experience, and previous experience and bias. We need to think about what we're going to talk about, the content, what are the key messages. We'll try and keep those to the point, the context of what we're telling them, decide what facts to share with people, and also how to collate opinions from people. We have to consider the purpose, the why we're doing this. Are we trying to explain theories? Are we trying to explain what we do as researchers or as academics? To provide a transparent background? Are we trying to give people skills so that they become competent in a particular area? Are we trying to provoke interest and excitement, which I hope we always try and do? And then finally, the how. What's the platform that we're going to be using? Is it going to be online? Is it going to be through written media? Where are we going to deliver this content? How much interaction can we have with the audience, whether or not they're able to participate, and things like language barriers. So taking those a little bit further, I want you to introduce you to the typical family. This typical family, of course, is very well known to most people. This is the Simpsons. We have Bart, Marge, Lisa, a little one whose name escapes me at the moment, and Bart. They might attend a mycological event and they have diverse interests. Possibly their interest has been developed because of this little tiny mushroom growing up from the floor in the corner of their room. So thinking about the who in the audience, well, there's Homer. Many people know Homer's key characteristic. He is strongly influenced by what's going on in this part of his anatomy, and is primarily interested in beer and food. So with Homer, we can start exploring stories about fungi related to beer and wine production and alternative fermentations. We can think about foodstuffs and the processing of foods by fungi. We can think about the importance of fungi in bread production or cheese production, or the production of chocolate, all key things, okay? So a starting point might be a discussion about the history of Saccharomyces pastorianus, which was formerly known as Saccharomyces carlsbergensis, definitely a name that I favour in terms of remembering its importance in brewing. And you can talk about yeast biology with Homer. The other who, of course, is Marge. Marge spends quite a lot of time in the kitchen. We're not stereotyping here, but that's the way she's portrayed in this particular image. And she might be interested in the food value of fungi. And she might have that as her primary aim. You can start off by talking about recipes for different mushroom dishes. Or you can explain to her the differences between different fungi, some of which are edible and delicious, and some of which are highly toxic and poisonous. In this pot here, we have a mixed bag of morels. We have the true morels, and we also have a false morel. So can you explain the difference to Marge between these different species? And potentially we can explore why Gyromitra esculenta is actually toxic. The fact that it produces a toxin called gyromitrin, which is broken down to monomethyl hydrazine, and even explore a nice little story about how we know that. The original work that led to the identification of this toxic compound came from work that was being done on the space shuttle in the 1970s and early 1980s. And it turns out that monomethyl hydrazine is used as a propellant in spacecraft, and many of the workers who were putting together the propellant material succumbed to seizures and behavioral issues, which led to people noticing the similarity with people who'd been poisoned by the false morel. Another who, this is Lisa. Lisa, of course, is the A-grade star student, and I managed to find a nice picture of her looking down a microscope. So we could introduce fungi to her through considering the biology of penicillin. Talk about antibiotic production, talk about human fungal diseases, for example, which is my current focus in the MRC Centre. Another who, of course, is Maggie. I struggled a little bit more to find out what Maggie was interested in, but this image here shows her with her nappy on, and babies experience nappy rash, and 80% of cases of nappy rash are associated with a Candida albicans infection. So again, it's an opportunity to discuss the biology of the fungi, the therapeutic options that are available, and the production of antifungal drugs and their origins. Finally, of course, we've got Bart. It didn't take me too long to find a picture to suit Bart's interest. So obviously this is the highly, the well-known phrase, "eat my shorts" from Bart. And here we've got a nice picture of a giant puffball, Calvatia gigantea. And you can talk about, you know, the importance of gastromycete fungi, talk about puffballs and stinkhorns, all sorts of things which get someone of art's sense of humour interested in the subject. So continuing on with the who, actually, I started to think about how we communicate mycology in response to the publication of a document by the World Health Organization. This document is shown down here. It was released about 2 years ago. And in this document, they highlight the importance of human fungal diseases. They give some amazing figures that 2 billion people a year experience a fungal infection. 1.5 million to 2 million people die every year from human fungal pathogens, and that level of mortality exceeds levels from other pathogens such as malaria or tuberculosis. It highlights the fact that in sub-Saharan Africa, many patients with HIV that isn't controlled succumb to fungal diseases such as PCP and Cryptococcus. So messaging is really, really important, and being able to get across that message in a clear and unambiguous way is really, really critical in terms of spreading a message. And of course, we have to think about the audience. Are we trying to talk to government agencies? Are we trying to talk to grant funders, public health agencies? Are we talking to students in universities, colleges, and schools? Are we communicating with special interest groups, patient groups, local foray groups? Are we at a science festival where the general public is there, who might have an interest in science but perhaps haven't thought about mycology? And of course, sometimes the who is your family. Trying to explain what you do for a living can sometimes be quite challenging. So we have to think about this audience really, really carefully. We sent out a questionnaire. I think there were 18 questions to the British Mycological Society membership about 2 years ago, and this is just a little snippet of the outputs from that. And one of the questions is, which of these best matches your current level of fungal knowledge? And we had choices from absolute beginner, beginner, advanced beginner, intermediate, advanced intermediate, giving some examples of who might fit into that category. And actually, most people who responded were advanced beginners. Now, we have here a very biased survey because it was sent out to members of the British Mycological Society. But it did give us some idea of what sorts of things that people were actually interested in. And people want to know more about identifying fungi using microscopes, and a little bit about the ecology of fungi. That came high up on the list of priorities. In terms of the why it's important to explain about mycology, then clearly we can build on the passion that many people have. Passion for the beauty of the organisms, passion for their uses as foodstuffs, passion for their ability to solve world problems through drug production, whether that's antibiotics or antifungal or antiviral. We also want sometimes just to entertain people, and I make sure that in my lectures that I do have elements which are a little bit more amusing, and maybe go away from the core message just briefly to explore bizarre facts and figures. We might just want to educate people for professional reasons, and provide training. And of course, in my role as an academic, we're always chasing after the money to secure our next research grant. So again, we need to be able to communicate the science effectively. So here's just a reminder of what it is that people want to know about. They want to know about fungi and their part in the natural world, fungal ecology. Actually, 71% of people who responded to our survey wanted to find out more about that. 87% of people were interested in finding out more about identifying fungi. And actually quite a high proportion, more than I thought, actually just wanted to be able to describe fungi and get to grips with the terminology. And of course, mycology is a very terminology dense subject. So what can we do to get people engaged in mycology and listen to what we have to say? And I think the important thing is having a narrative story, having something that people can follow, they can relate to, and that they intrinsically find interesting. So you can use as a starting point something like this book, What Moves the Dead. You can talk about the benefits of fungal sex. I've never found any audience that doesn't get interested when you start talking about sex in fungi. Or you can talk about specific groups. So here we've got a representative image with lots of different gastromycetes, earth balls, puffballs, stinkhorns, this cage fungus, and so on. And so we can try and tell stories based on these different topic areas. So here's my first mini myco story. This is a topic which has become widely of interest because of the television series The Last of Us. In The Last of Us, the tale is about zombies and the colonization of hosts by a cordyceps fungus, possibly based on Ophiocordyceps unilateralis, and the changes in behavior of the hosts that are associated with those infections. I've had a long-standing interest in this particular fungus, Entomophthora muscae. This is an entomophthalalian fungus. The name is the clue. Insect slayer is what it translates as. And domestic houseflies and other insects could become colonized by this fungus when a spore lands on the surface. The spore then produces enzymes that allow it to penetrate the cuticle of the insect and get into the hemosyl. The mycelium then spreads throughout the body and ultimately leads to fruiting structures here. What's interesting, of course, in this particular image, sex always tells a good story, is that these dying hosts are still attractive to other non-infected insects. And so we've got a manipulation of the host behavior. We've got potentially the production of drugs, which we don't know what they are, that can manipulate host behavior in some way, and the transmission of that lethal disease. Second mini myco story I want to explore is actually related to what I did during my PhD with Alan Rayner back in the late 1980s and early 1990s. I chose to work on signaling systems between different individuals within a mated mycelium. And the story that got me hooked on this was actually based on this particular image. This is taken by Martin Ainsworth, who was a postdoc in Alan's lab at the time. And it shows two strains of a fungus, or two species, in fact, Sterium complicatum and Sterium hirsutum, that have exchanged genetic material. Nuclear exchange and nuclear migration is taking place either side of this mating response. And we've got the establishment of two heterokaryons, one on the complicatum side and one on the hirsutum side. And what we see is the appearance of a phenotypically uniform, perhaps some would say somewhat bland mycelium on the complicatum side. And on this side here, where we've got Sterium hirsutum, we've got zones of instability, we've got deterioration and degeneration of the mycelium, as well as mycelial tufts and outgrowths, which seem a little bit happier. The story behind this, of course, that I'm perhaps hiding, people have probably forgotten, is that this is an American strain and this is a Russian strain. And it's about the expression of information in these two different geographical zones within the mycelium and the consequences of that. So that particular story triggered a fascination for mating responses and genetic exchange systems in fungi, and conflict resolution as well, which was the topic of my thesis. So here's your chance now to explore your own, or tell me more about your own myco stories. What are the favorite things that you like to tell people about fungi? Is it something about their biology? Are there particular stories that you like to tell? If you could share those with us, then we could explore those a little bit later on. Okay, finally, the how. How do you approach an audience and what do you do? Well, lectures and practicals are my bread and butter, I suppose. I teach large classes of up to 400 students in various aspects of microbiology, focusing on mycology. I have large practical classes with 200 students at a time. That is a very well established format. And the important thing really is to make a link between the hands-on activities and the theory. We also have the web and the social media opportunities. Can we use that to promote a better understanding of fungal biology? As part of UK Fungus Day, I've become much more familiar with social media platforms than I was previously. And last year we were releasing fungus facts, in fact, fungal financial facts. Here's a little known fact. The global value of fungi to the world economy is estimated to be around about $54 trillion or 35,000 trillion pounds. And the contributions that yeast plays within that are larger than the GDP of Australia. So alcohol production is very, very high up on that list. Okay, so we need to think about the how, whether we're going to use web and social media, traditional media. Are there opportunities for forays, school visits? Can we use games, for example, to aid in education? Can we set up some exhibitions? And are there any contributions that we or you could make to UK Fungus Day? Last year was very busy for me as the chair of the committee and having a very close role in organising UK Fungus Day initiatives. But in my role within the MRC Centre for Medical Mycology, I was also involved in lots of other outreach activities as well. We do have a dedicated outreach team, and so that helps a little bit. But we were involved in Soapbox Science. Here's pictures of some of the students and actually some of my colleagues. This is Liliane Mukamira here down on the quay in Exeter, explaining about Cryptococcus to a wider audience, an unsuspecting audience. We set up a superbugs exhibition by taking over an empty shop unit in the centre of Exeter. We've had the Killer Fungus Initiative, building on what had been done previously in Aberdeen and Manchester, and ran that in Exeter as part of the British Science Festival. For UK Fungus Day, I had an open doors event where we got all the researchers together from Exeter to showcase their research and tell stories to people about what they've been doing. And also we had a film compilation that ran in a local cinema in Exeter, and that was packed out with people. So we're just going to talk about a few of these things as examples of what I've been doing and what hopefully might inspire other people to get involved with education. So gaming. I've noticed over time that games are a really good way of getting people involved and engaged. We all know how easy it is to be drawn into a game and become very fixated on it, and as a result perhaps learn things that we wouldn't otherwise have done. So with the BMS and the MRC Centre for Medical Mycology, we put together this Fungus Facts card game. It's based on a very well-known Top Trumps style game. We can't call it Top Trumps for obvious legal reasons, so we call it Fungus Facts. But essentially there are facts around the mortality rate, the prevalence per 100,000, the morphological form, and the susceptibility of these fungal pathogens to various antifungal drugs such as amphotericin, fluconazole, as well as publication number. So with an undergraduate student, in this case Sasha Slater, we put together this pack of 36 human fungal pathogens, and you can play this game for fun, obviously, play it as a 12-year-old, but playing it with different age groups actually allows you to explore different things. So I played it with 12-year-olds, 17-year-olds, A-level students, and university and master's level students as well. We've also more recently created a plant pathogens version of this, so Amelie Ward helped put that together as well. So if you're a 12-year-old, you can just focus on the cool and bizarre names and some of the gruesome statistics around human fungal pathogens. And ultimately, you know, it's, do I win all the cards or not? 17-year-olds thinking about, you know, what excites them in biology. We can talk about which different groups of fungi are involved and the fact that there's more than one. We can ask questions about which pathogens are more lethal. We can actually talk about what an MIC is, or minimum inhibitory concentration of a drug, what we mean by the term dimorphism and why it's important. And again, playing the game, do you win all the cards? And as a 21-year-old or more, so this is what I've been doing with my master's students on my online course in medical mycology. We actually look at the data, and I get the students to produce their own cards. I give them a list of the pathogens and say, make your own cards, go and find out the information. And then we play the game online, and I compare the data I have on my cards with what they've got on their cards. We talk about the sources and the reliability of that data that underpins what's gone onto those card designs. And then that promotes a discussion of things like why antifungal drug resistance levels are variable between different species and different reports of the same species. Another area where we've been using gaming to look at fungal education is in teaching core skills to undergraduate and postgraduate students. We have at Exeter a long-standing collaboration with an international group called Labster, and they produce virtual simulations of laboratory environments. The idea is that you can expose students in a safe online environment to very complex pieces of kit. So we actually had the Labster team visitors. They took pictures of our confocal microscopes and then recreated those in a virtual environment. And in order to make the learning more exciting, we actually have a narrative, and the narrative is based around this guy here. This character is a farmer who has a wheat crop, and it's infected by Mycosphaerella. So we were able to simultaneously train people in how to use a confocal microscope correctly, but also get them to look at fungi. So here you can see some of the images that we've taken with the confocal, showing the development of a fungal hypha and septa and so on, and explore a story around that. And we're putting across some mycological content in this online gaming environment. Another thing that gets people excited is anything that kills you is going to be of interest, or kills other things is going to be of interest. So in the UK, there's a famous television series called The Deadly 60. And that has got people's imagination captured at lots of different age groups. And we thought, could we do something similar with the British Mycological Society? So we brought together some little tiny descriptors of 40 frightful fungi. And on the website that we had set up, you could click on the various images and find out a little bit, some gruesome statistics about a whole range of different fungi, from those that are poisonous, those that cause disease in humans and in plants and animals, to amphibians down here and so on, and in other fungi as well. So that was a resource that we created for the BMS. I mentioned superbugs earlier. This was an opportunity to put up a very highly professional stand showcasing the research that we do at the MRC Centre for Medical Mycology, but also get people involved a little bit in citizen science. So my particular contribution to this has been me and my mycobiome visitors to the centre, visitors to our UK Fungus Day exhibition for the open doors, or to the superbugs, could take swabs from behind the ear or from behind your watch. These were then streaked out onto selective media, and the different fungi that were produced were then photographed and then sent back to the visitors, and that was superimposed onto an image of them holding a plate with them and their microbiome. And this has actually turned out to be a great exercise, because we can start getting people involved in surveys, revealing information about their healthcare regimes, looking at antifungal drug resistance and so on. Okay. I'm conscious of time. I think it's 8:20 at the moment. I think I've got another about 10 minutes or so. So another thing that I've explored this year, and this is the first time that I've done this, I've led lots of forays in woodland, and it's very easy to engage people with real material, show them the fruit bodies, show them the effects on the environment, the wood decay and so on. But that's not always possible. People live in different environments and can't necessarily access those. So as part of a science festival that was held in Cornwall, I led my first urban walkie-talkie. And the idea was that we would take people for an hour through a stroll through the high street and talk about fungi. I don't know if people can see on that image anything that might jump out at them as being a relevance in terms of mycology. But I managed to find a few things that provoked conversation and discussion. So this is Falmouth town centre. And so I've zoomed in on that a little bit more. Of course, in the town centre, we have people. I've just chosen this chap down here. I apologise to him if he's in the audience. I think it's very unlikely. But we can talk about Malassezia infections and the contributions that they make to dandruff, focusing on my own particular interest in medical mycology. We have a pub, so we can talk about brewing and Saccharomyces cerevisiae biology. I'm losing track of where we are now. Shoe repairs, role that fungi might play in leather production, leather decomposition. We can also talk about this. I think this is a restaurant, role of fungi in food production. So there are all sorts of opportunities to spend 5 minutes, 10 minutes outside a shop talking about a mycological story. So a bookshop is useful in terms of talking about biodiversity. A typical bookshop contains about 3,500 books, each with about 300 pages, which gives you about 900,000 different pages. And you can talk about the fact that you couldn't even describe all the fungi in the world on each one of those pages because there's so many of them, and talk about the diversity of fungi. You can talk about the role that fungi play in manufacturing processes, such as the production of paper. At the bakery, we can talk about yeast, genetic engineering, cold tolerance. Go to the pub, talk about the power of yeast again, Nobel Prizes for the cell cycle or for autophagy. My own particular research at the moment is focused on animal models of infection. And so going to a fish shop or a fish and chip shop allows me to talk about fish embryology, fish immunity, and the interaction with human fungal pathogens. We can go to a clothes shop, talk about fabrics and conditioners, talk about textile manufacturing. In Falmouth, I found an art shop that had a wonderful autumn display. So talked about leaf litter decomposition. We went to the grocers and talked about food production and food security, focusing on stories around pathogens and mycorrhiza. A furniture shop looking at wood production, wood decay, and beautiful forms of decaying wood. We even visited Vision Express, which is obviously a place where you can buy spectacles. but their key character at the moment in their adverts is a mole. So I was able to talk about mole latrines and Hebeloma radicosum, which is a mushroom that produces pseudo-rhiza that connects to the nitrogenous material in mole feces and waste, but also allowed me to talk about eye infections and the role that fungi play in those. We visited a churchyard and talked about Hebeloma venosiphilum, which is a mushroom that grows on corpses. And also you can usually find a yew tree, so you can talk about taxol production as well. And at a pharmacy, we can talk about drugs, antifungal drugs, antibiotics that are produced by fungi and human fungal diseases. So all sorts of different things that you can do in a different environment. If you're a researcher, then I would encourage you to do an open doors event. We invited people into the University of Exeter, both in 2022 and 2023. We had around about 600 visitors with very little advertising for that particular event. It coincided with UK Fungus Day, and we were able to explore in our first Open Doors event purely medical mycology, and in the second Open Doors event all of the other mycology that's going on in the University of Exeter. As a result of that experience, we put together a set of guidelines, or at least a checklist of how you can organise your own Open Doors event. So it covers the planning, the timelines, what permissions you might need, the health and safety, the communications, what's most effective, what's not so effective, and what to think about on the day. Everything from making sure that the toilets are well signposted, tea and coffee is available, to making sure that all the displays are in the right place and looking great. So here are some of the examples of things that we had. We had animal models, building your own cell wall, fight the fungus, finding the hidden link in relation to DNA sequencing, from ions to biomes, looking at the relationship between the physiology of fungi and the landscape, fungal art. And we've done some great things from all age groups, from preschool right through to professional artists coming in and showcasing their work, playing the fungus facts game and so on, playing immunology Jenga, talking about me and my microbiome, demonstrating point of care tests and the science that's gone into development of those, and having over here you can see an art installation produced by Leonie Hampton. She spent 12 months on a residency with us in the centre and produced this amazing bronze sculpture showing Candida albicans hyphae as they grow through a blood vessel and interact with macrophages. So she was able to explain to the public the significance of that, what the inspiration was. It's also really useful to have a go-to talk. I have a talk called The Wonderful World of Fungi. It explores fungal biodiversity, fungal cell biology and physiology. It's got sections on biotechnology and bioremediation. It's got stories around foodstuffs, poisonings, all sorts of different things. And it's a collection of around about 60 slides, which at very short notice I can usually put together and build a story from something that will last 20 minutes to something that will last an hour and a half, depending on the time that's available. It's also good to have alongside that some go-to activities. So here's me at an outreach event last year. It's a BMS stand and an MRC stand. Got photographs of fungi at a very small bench space here, just a small rickety table with a microscope and some cultures. Lots of leaflets from the BMS and from the MRC centre. But the most important thing is to find out what it is that people are interested in and talk to them about it. And actually, as a result of that, I realized there was nothing on the table that appealed to preschool children. So fortunately, I was able to quickly come up with fungus Fred. This is based on blowing up a hand glove, laboratory hand glove, getting the kids then to draw faces on it. And actually this led to something which we did at UK Fungus Day, which was fungus Fred, fungi, fungal, or fungus. And it allowed people just to make beautiful characters, but you can also see, you know, how this could be used to explain about conidiogenesis and blastoconidia, for example, or different conidial shapes and how the morphology is regulated, the role of turgor pressure in determining cell shape, as well as talk about the ambiguity of the concept of sex in fungi with the fungi, fungal, and fungus initiative. I also have, as a result of the undergraduate programs that I do, boxes full of different topic areas related to mycology. So here's my foodstuffs box, which I can just bring out and take to events. So what I want to do is encourage everybody who's in the audience to get involved. If you are very interested in joining us on the BMS, we have a number of FEO working groups. We have a newcomers group, an education group, and a UK Fungus Day group. I believe that Sally has already sent out some emails asking people if they're interested in that. We are currently reviewing the BMS website. So if you have any suggestions for the improvement, either in content or the way it looks, please let us know. We're trying to think of new resources. We've just undertaken, excuse me, a review of the existing resources we have. We have around about 50 different packs that we can send out with information on, and tell us about how you think we can engage with new audiences. And please do take part. Please do get in touch with schools, universities, get involved with public engagement. If you're an artist, we have the Massey Arts Grant, which is currently being advertised at the moment on the BMS webpage. There's a significant amount of funding available, £5,000 for 2 different projects to run in 2024. And as long as that can be linked into UK Fungus Day in some way, we'll be very happy. So please do get involved. So I just want to finish by acknowledging all those people that I've already mentioned from my very formative years as a child, and then as a student, and now as an associate professor leading the training for the MRC Centre for Medical Mycology. So thank you to all those people. This is just one snapshot of the people who were in the centre last year at our annual retreat. So thank you for your attention, and I guess I need to stop screen sharing now, and we can take a look at the chat and see what's there.
Thanks, Mark. That was super, absolutely fascinating. There are loads of things in the chat, and I can read some of them out to you in a minute with regard to your questions. But I guess now might be a good time also to mention to people if there are any other things they'd like to ask. Perhaps they would like to put those in the chat now. I can see things coming up already, but we'll turn our attention to those in a minute. Perhaps if I tell you first some of the answers that came to your question, who were your influencers? And they fall into all sorts of different categories, and I've tried sort of to categorize some of them. So the first ones, which came shooting in on the screen, were really people who are, I suppose, field mycologists and people who've written books about identification and fungi in the field. So Richard Forty Brucing, Jeff Kibbe, Roger Phillips, Debbie Evans, and several people in there get several mentions. Another couple of people mentioned their father, so that was nice too. Of course, lots of people mentioned academics, such as Alan Rayner, Tony Lyon, Roy Watlin, Gareth Griffith, also from the other side of the North Atlantic, E.O. Wilson, the great E.O. Wilson, John Peabody, Harry Hudson, and David Ingram from Cambridge. also other people actually from overseas, Ramsdale Gallis, and the great Juliana Furkey with Fungi Foundation, natural history societies and teachers, so people like Jim Cook of the Dundee Naturalists, Kathleen Roberts, Wakefield Naturalists, the Edinburgh and Lothian Fungus Group. Then there were mention of food people, for people who run food courses or food books, such as Marlowe from Wild Food, Antonio Carluccio's books. And then other people also mentioned books and authors and broadcasters. So Gerald Durrell, who you've already mentioned, Langan Hora, a book called In Search of Mycotopia, Merlin Sheldrake's recent book on entangled life.
I was going to say that I think we are in a renaissance, if we can have a renaissance of mycology at the moment because of that particular book. You know, it appeared in the Times top 100 bestseller list. It was available in airports. Obviously, I'm fortunate enough to be able to travel around, and you go into an airport anywhere in the world and you'll find it there in their WH equivalent, and I think that is absolutely amazing. And it's full of stories, and I think that's why it's so engaging.
Yeah, I agree. Great book, beautifully told, and the first time fungi have ever been hyped, I think. So yeah, really awesome. So all sorts of different people there. I mean, guess this is the sort of people you'd think, but nonetheless very fascinating, I think. I don't know whether you've got any comments on the categories of inspiration that I gave, or whether you'd like me to move on to telling you some of the answers to some of the questions.
It's clear that an awful lot of different people have inspired the audience, and I guess it's just taking that opportunity to identify that point of ignition, and you know, all these different inputs have led to people getting excited about fungi. So the more little flickers there are out there, then the more people will take notice.
Absolutely, and I can see even more names have been coming in since I noted those down.
So what about the particular fungal stories, though?
Yes, so there's a range of different fungal stories. Obviously, there are some repeats, but I'll read some of them out to you. So the first one which came in was Robigo, the Roman goddess of rust. And again on pathogens, ergot, impact on humans, LSD, witchcraft. Massospora curcudina, so like your story of Entomophora musci. Quite a few people mentioned mycorrhizal fungi, communication, collaboration with trees, things like that. Scomanita paradoxa, parasitizing cystoderma, fungi in Chernobyl, different ways fungi can kill you. So I think people are a bit like you there, Mark, and agree that the deathly things are quite fascinating. Physarum polycephalum, well, that's a slime mold, but we'll let that off, because if it gets people into real fungi, that's great. Quite a few mentions of diversity. Pilobulus, like you, yes, that's always a great one. I'm up for that too. Rust fungi life cycle, mycoremediation. Several people mentioned dung fungi again. They're always great fun, aren't they? Fungi on sculptures, pseudosclerotial plates. There's a fun one. I could have written that myself. Flyogaric mythology. And I should put in a plug here. Diana Richards from Fungi Foundation mentions that to remind us all that there are free resources for children, telling children about fungi on their website. So let's have a big plug there for Fungi Foundation. And a question actually which came in whilst I was noting these down, which I thought was an interesting one, I don't know the answer to. Is there a scout or guide badge for fungi?
This is something that we've talked about at the FEO, and we thought that maybe there should be one, but we're not aware of one. There's a natural history badge, but nothing specific on fungi. And I think Gary, who might be online.
He is online. I think he's got a question coming in somewhere down the list, I think I've noticed. I think we might. Oh, here we go. Let's have one from Gary, as we mentioned his name. Regarding the FEO committee, what has worked the best and what hasn't worked at all? That's a question for you, Mark. Strong points and weak points.
I think what works best, the Open Doors event has worked really well for us, but we've run it in Exeter twice now, and it would be nice to see that extended to other centres of mycological research, and there are many within the UK, so that's really something that we would like to see expanded. The film festival worked amazingly in Exeter, and we also had it running online for one day only, which was quite nice. But getting that out to mainstream cinemas, we were going to give it to them for free, no cost to them, and nobody took it up. I think the Welsh screening agency, I can't remember their name, the UK split up into different quadrants in terms of film releases, and Wales said, oh yes, that sounds interesting, and then never followed it up in terms of release. We have struggled to get business engagement, so we contacted, I think, about 30 different businesses that are involved in fungal work, mushroom composting companies and various things, and got zero responses from anybody on that. And so there are certainly some parts of the fungal equation that aren't publicizing what they do particularly well at the moment, and they're obviously too busy. There are cost implications, there may be IP issues, all sorts of things, and it would be nice to sort of get a little bit more involvement from those.
Yeah. There's a question here. Do you have recommendations for books on medical mycology similar to Blight by Emily Monosson?
Do we have, I'm not aware of any accessible books. I don't think you count the Oxford textbook of medical mycology as being an easy read. Definitely an opening in the market there.
Yeah. Here we go. Here's a nice one. Can mycology take over from dinosaurs in the imagination of youngsters?
Well, the death of dinosaurs can be taken over by mycology, if you like that particular narrative story.
Yeah, I suspect they might have been thinking, can we, in the same way as young kids will go for all these dinosaur books with all these great long names, can we do the same for fungi? Let's hope we can. Actually, I'll come back to check on that in a minute. In my opinion, it is very important to capture the interest of young children in fungi. And I was wondering if you have any comments or ideas on how to develop educational resources for elementary school children.
So that is part of our review plan. We're currently focusing on secondary schools, but my wife is a primary school teacher, and it's, I was going to say, it's very endearing. It's just wonderful to see the enthusiasm from small children. We have some lovely painting activities based on using pipettes to move brightly coloured fluids around and to decorate and produce their own fungal images. We have modelling activities in which they can create their own fungus based on images and stories that we've told. So those things can work quite well. But I must admit, the younger the children are, the more scary they are to me personally.
Yeah, a good one. So there's one here, which is really, I suppose, for me, actually. Wanted to ask, very complimentary about the BMS symposium, wanted to ask if anyone can recommend any books on decay and regeneration, interested in the idea of rot and rebirth. It says, I'm an artist, not a scientist, so find it difficult to read journals. I don't know whether you could answer that one, Mark.
Again, I'm not specifically aware. That's one of the reasons to go to a bookshop on my tours, is that you can always introduce people to that relevant section in the bookshop. I don't know is the answer, and that's also something that's really important in education, is that there's a huge amount that we don't know. There's a huge amount unseen, undiscovered, and that's the excitement. That was one of the things that Henri Descalzes introduced me to when I was 12 years old, was the concept that I could be looking at new species that had never been seen by science before, which as a child is amazing. Still is amazing.
Yeah, oh, absolutely. Yeah, I think that every day. I think, yeah. I mean, I think
I have seen one comment on there from Caroline Hobart, and I think it says that you're missing the fact that most of your events are run by the groups, and I am immensely indebted to what the groups do, and that's why we've made sure that we do get a good interaction with the local groups. We had over 40 different regional groups take part in UK Fungus Day last year, and looking at the statistics, we think around about somewhere between 5,000 and 10,000 people took part on UK Fungus Day, and I think that's, you know, down to the local groups and local organisers.
Absolutely. Here's another one which I really agree with. As far as education and outreach is concerned, surely school children must be a priority. I lead lots of forays for local wildlife trust, but my wife points out that I'm just preaching to the converted and should be directing my efforts towards children. I couldn't agree more, and being involved with writing a children's book at the moment with Ali Ashby, who's also on this call. I mean, I think we'd agree with that. But maybe you'd like to say something too.
I absolutely, and one of the committee members on the FEO is Christian Taylor, and I say he's got lots of experience as a school teacher, and he's done an evaluation of all of the different GCSE and A-level curricula from all of the different examination boards, and highlighted where fungal biology is present in the curriculum, because it's very hard to introduce something into a school if it doesn't relate to the curriculum, and has also suggested various activities where you could bring fungal biology into a particular story, and I say you can do it surreptitiously that way.
That's excellent, because that's also that's answered a question by Annabelle Lambert, which is in the chat, which I won't read out. I just remembered that I spotted something earlier on when I was going through pulling out these lists of what are your favourite topics, etc. Someone was asking about the new natural history GCSE and whether fungi will be involved in that. And yes, they most certainly will. We have representation on those committees, but as yet we don't know in what way fungi will find themselves woven into the framework. But I'm on that committee and I hope that they will get there. Some more things coming in. I'm just reading these straight away. So some public and subscription libraries have small cinemas and associated film groups. That comes back to your other comment. Oh, it's been tricky to get public schools to adopt more fungi education into their curriculum, but that's what we're working on too. In addition to our current lessons, we are working on comprehensive curriculum aligned with next generation science standards, so more schools will adopt the curriculum. Excellent. Oh, here we go. There's one on Merlin Sheldrake now. Merlin Sheldrake, he does indeed, has a film coming out on IMAX big screen cinema called Fungi Web of Life, narrated by Icelandic superstar singer Björk. Since there will not be that many screenings, I know of only one scheduled so far in the UK at the British Film Institute in the South Bank in London. Any chance of leafletting or asking the BFI to allow BMS to have a stall at these events?
So I guess there are two issues there, or two questions wrapped up. One is, can the film be shown in more sites? In small arts house cinemas were our particular market target. And I say we were offering 40 minutes of entertainment for free, and nobody wanted to take it up. Maybe if we charged, that might have got more people to want to come along. But I say in Exeter, we sold out the free tickets and filled up the cinema. So it can be done. Similarly, you know, I contacted every university cinema club in the country and got no response from any of them, which is, you know, I've been a bit taken aback by the apathy within, you know, some of the communities that we're trying to do stuff for. But that doesn't mean to say we shouldn't stop and that we should continue trying.
Yeah, yeah. Oh, I like this one. My 6-year-old grandson is almost as obsessed as I am and loves looking through the fungi of temperate Europe. He wants his own copy. Well, that's good taste there. Excellent taste, I would say.
I'd quite like to look through the fungi of temperate Europe as well, but not in the book. Just spend the time.
Yeah. Let's see what else we got. Our children are the next generation of mycologists, so important to engage with them. Couldn't agree more. Oh, there's the natural history GCSE coming up from Gary again. Someone recommending The Lives of Fungi, a natural history of our planet's decomposers. Oh yes, I'd forgotten that. I have that on my bookshelf. Thank you for mentioning that. Here's another one I'll read out. I ran an art exhibition with sculptures made of different substrates and grain spawn plugs instead of clay. The show generated great response. I want to create another art show displaying native and local fungi isolated on Petri dishes. Sounds good to me. Yes, I agree with this. When are we going to get ecologist reports mentioning fungi? Well, I think we've been drip feeding for the last goodness knows how many years, and I think these things perhaps are just about starting to happen now. You know, thanks to Fungi Foundation and Juliana Furkey and others, we have flora, fauna, and funga mentioned. Now noted by IUCN, and that's a huge step in the right direction. And various governments starting to take notice as well. Oh, here's somebody says you can order the film to a local cinema. I live in Cumbria, so only have an independent cinema. Okay.
So we did manage to get our film shown at the Armet Museum in Ambleside. That was the only other place other than Exeter that took the offer up.
Right. Here's another book mentioned, Let's Become Fungal, a really great book. I like this one. Do we have a national mushroom? If not, I think this would be good, like a national flower or animal. I think it would be good too, and I'm sure we don't have one.
I think we could debate for a long time, though, as to which.
Yes. Yes. Oh, it's just a little advert for Merlin's Fungi Web of Life film, 9th, 10th and 11th February, BFI IMAX Waterloo. Comments on the natural history GCSE, really important. It is cross-curricular and includes arts and humanities. Yes, I'm not sure that to what extent it will include arts and humanities. That was mentioned, I believe, when we had the last discussion. So let's hope so. But it's certainly going to be cross-cutting, including fungi will be included along with plants and animals. Oh, I think I've come to the bottom of the list now.
Okay.
Wow. I don't know whether anybody else has any last questions, but I think if not, that's actually rather perfect timing. It's almost 9 o'clock, so not far from my bedtime. So I'd like to thank you once again, Mark, very much for a great talk. It was really great to hear your ideas, and also to thank the audience very much. We had over 60 people, and many, probably most people, commented in the chat. And came up with these ideas of their favorite things and who their influences were, and that's really set me thinking. So I think it's great, and we've had a very nice discussion for the last half hour. But I don't know whether you'd like to say anything finally, Mark.
No,
loads of thanks in the chat there have been.
No, well, I thank everybody for their patience in listening to me ramble in a reflective way about education. It wasn't something that I thought about presenting before as a topic in its entirety. So thank you very much for sticking with me for this time. And please, everybody, go out and share your knowledge, passion, and enthusiasm with people that you meet.
Well, it's Mark, just one thing that's come in. Could you share your PowerPoint on the BMS fungal biology web pages?
I believe that we are recording this session, and I guess after a little bit of editing, I'm sure that Sally would be able to make that available.
Excellent. So on that note, thank you once again, Mark. Thank you, BMS, as ever, and thank you, audience, and hope to see you all again soon. Bye-bye.